Okay, I think this is started. Video. There we go. Okay. So I'm going to repeat a couple of things that I just told you guys. You may have one, shouldn't show a whole stack of them, one index card. You can use both sides. Okay. One index card must be 3 by 5 inch if you needed to either find one on your own because you're not here for this in-person session. So 3 by 5 inches, I think, whatever that standard sort of size is. If you bring one in and it doesn't like match my footprint, that's going to be a problem for you. So make sure that it matches the standard size index card. So you can bring that to the exam. You'll turn it in with your exam. Okay. Think, and I will go through sort of the review or the review sheet in a minute, but think sort of high level stuff, but sort of integrated. There are a number of tables that you have to fill in. It's asking you to sort of like pull together information about things that we maybe have learned as individual learning units, but I want you to be able to see the high level integrated connection.
Like that, you know, like our homework several on tables like that, or is it going to be like putting those tables?
So, as an example, thinking about something like all of the different things that we learned about the 4 major biological molecules, right? So what's their caloric value? What are the atoms that make up them? What are the linkages that you have? What are the monomers called? You know, all of these different, are there any special numbers that we had? Or is there an important molecule that we learned about? So sort of like can you assimilate and think about that 30,000 foot view knowledge? I will say too, there is a mechanism on there. Sorry, it is. No, we didn't get, you should be able to do this mechanism. If you cannot do this mechanism, then we have completely failed as organic chemistry and biochemistry. Is what? No, you won't fail the exam. It's not, what's that? That exam or that, I don't know. It's not that much. I will say there is a big table at the end, which asks you to sort of think about all of these different metabolic pathways. So for each metabolic path, like when we learned about glycolysis, be able to think about like starting and ending metabolites, who's the rate controlling step. Sort of think about fundamentals levels stuff for that. Is it a need state or a fed state pathway? So those sort of things. So as you're studying those, like you do not need to know the entirety of glycolysis as we ran it through here, but there are certain pieces that we continue to sort of think about. I'm going to pull up, and I'm not going to share my screen here because you guys can see, well, some of you have computers, just be able to see that. Maybe I'll share my screen here in a second. I was going to pull up the study guide that we have here. And I will say the study guide is kind of like it looks behemoth because it's like, wow, that's like the entirety of the course. But I do expect you to know your amino acids, right? So there is a math question. Buffers are really important in biochemistry. So I would know from lab math how to make a buffer. Okay. Henderson-Hasselbalch equation. So there were some equations that you sort of needed to know, whether they were Henderson-Hasselbalch, 2 thermodynamics equations, things like that. I think I have a schema. This will be easy here. I'll just pull up here as we're kind of running through here. Turn this off and we'll turn it back on. So as I'm looking at sort of the study guide here, know your basic fuel equation, right? We've expanded on that. So I'm just reading here from the study guide, like your basic fuel equation. And then we even expanded on that. I still hope that you'd be able to kind of understand, like, yeah, I know what the basic equation is, but how do we make CO2? How do we make ATP? Like things like that. There we go. I was not able to move things here. This should hopefully be connecting as soon as that finishes up here. Yeah, there is a reaction mechanism. It's not overly complicated. It's one you've seen before. So study all your old reaction mechanisms. It's not new. You have seen this one. Okay. But I hope that you'd be able to kind of put it back together. Acid-base chemistry, again, think of the Henderson-Hasselbalch equation, be able to tell me if I were to show you a molecule, like what the protonation state would on that molecule would be. What would that molecule look like at a certain pH based on the pKa values that you need to know and what pH it tell you you're at, right? Know some important numbers. Like, again, this is another one of those tables. It's like, hey, here's some important numbers that we've talked about. Like, think about things like physiological temperature and pH, blood glucose concentration, how long a red blood cell sticks around. Like anytime that we sort of had, and they're likely to not be these little esoteric things that maybe we talked about once and never revisited again. A lot of these things, we've sort of seen them time and time again. Functional groups, I still expect you to know your functional groups. Obviously, there's ones that we really haven't seen repeatedly, but a lot of stuff from Biochem 1 sort of became an applied thread in Biochem 2. We do know 2 thermodynamics equations. We do need to understand how equilibria shift. Be able to draw a reaction coordinate. Hopefully that's something that you understand, how to draw a reaction coordinate, how to think about how delta G double dagger and how delta G sort of play into that. Be able to rank your intermolecular forces and give me an example of where we might see them biologically or biochemically. You do need to know how to draw some molecules. Think about like the 4 things that we know about. You need to know how to glucose. That was our staple carbohydrate. Be able to draw a peptide, right? If I gave you a cute little word with some letters that you'd know what those letters refer to. For full credit, you know, hey, it would be great if you could tell me what those amino acids were, but for most of the credit, tell me the functional group that's there or draw the functional group that's there. Short little nucleic acid, you know, short little dinucleotide, let's say, be able to draw, you know, your nucleic acid backbone and so forth. So again, looking at these are just kind of running through. So being able to draw a basic nucleic acid. Obviously, we do not need to know how to draw these, but know what they are used for. Like, where did we see these? What's the B vitamins that they are derived from as they have B vitamin derivatives? Expect you to still know your 20 amino acids. Functional group reactivity. I don't know what I mean by location there. Location. I don't know what I mean by location. Relative pKa. Either 3-letter and 1-letter codes, importantly, the 1-letter code. Those of you who have done any studying with MCAT know that knowing your amino acids is like a really, really important thing. So that serves double purposes. Being able to think about peptide or disulfide bonds. We talked a lot about glutathione. And as I'm running through and just having a word vomit that I have with you here, if there's something that you're like, hey, can you say a little bit more about that? That's not like ringing a bell. This is not meant to be the deep dive. This is meant to be, hey, do you know what PI is? Right? Do we know what PI is? Say it again. Oh, that is not the right answer. Nope. So when we have P with a little sub I, so I was, yes, this PI. It's for proteins. It's like the pKa for a protein. It's the pH at which the protein has no net charge. So when you have something that has multiple ionizable groups, there's going to be some pH value where all the pluses and minuses will cancel one another out and you have no net charge. Okay. So again, as you're going through here, if you couldn't, and I know some of you study together, like try to have a little rapid fire Jeopardy thing like I'm doing right now. Like, tell me what PI is. And if you both can't answer that question, okay, we got to go look that up. Right. But it's high level. Like, we're not going to do any calculations with that or something. Glutathione, know what glutathione, you know, GSH is glutathione. What is glutathione? That was a question. That was not a rhetorical. That was a, hey, I'll play Jeopardy with you. What is glutathione? Those of you who did the alphabet soup, hopefully maybe had GSH as part of your, so glutathione is an important biological antioxidant, right? We saw it several times sort of throughout the semester. We saw it seen as an antioxidant in several places. We saw it with when we talk about Tylenol overdose, and it sort of gets overwhelmed there. It's a way that we can regenerate some important biological molecules. and so forth. Let's see, what do we have here? Not too much that you need to know here, but think about like anything that magic numbers were like 40 and 400, like, I'm sorry, 4,000. Anything less than 40 amino acids, you really don't have enough to have a structure. Anything greater than 4,000, it's a problem because you start making errors. And so those are sort of the brackets for where we see with protein sizes. And then we sort of talked about diversity in the sense that we really don't see, I'm going to zoom in here. We really don't see every possible option. Maybe those little math problems that it's like, hey, if you wanted to make a 10 residue peptide using all 20 amino acids, how many options are there? Right? So be able to do those types of, remember, it has to do with either factorial or exponents. Like if you had a 10 residue peptide, anybody know how to do this math? So it's going to, if you had a 10 residue peptide, you have 10 spots. So it's going to be 10, right? Or no, I'm sorry, it's going to be 20 raised to the 10. So there's 20 options and there's 10 spots. So you can think about it, if you ever wonder, like, how do I figure out that math? And that's why I had to do it in my head. I have 20 options for the first spot. If I can reuse amino acids, it's 20 options for the second spot. So that's 20 times 20. That's 20 to the second. That tells me that exponent is the number of residues I'm going to have. Like if I want to make 10, the 10 becomes my exponent. I have 20 options for what I can put in each position. So for nucleic acid, it would be 4 to the 10th. Right? 4 options for my nucleotides. So being able to think about little math things like that. Again, I already told you to be able to draw a short peptide. Thinking about this, how do we form structure, right? It's a complement of sterics and energetics. We don't want things banging into one another. So we have certain allowed phi and psi angles. That's what a Ramachandran plot was about. And then energetics, if we're going to make, that's an entropy-enthalpy compensation, right? If we're going to hold something still, what penalty is that? Enthalpy or entropy? It's an entropic penalty. So we better form some enthalpic compensation. Enthalpy, I always want you to be thinking about those intermolecular forces. We had one of those tables once to think about thermodynamics, know the letters, know the word, the single letter words, and then the phrases that we sort of had with those. Understanding the difference between how we make primary, secondary, tertiary structures for proteins, right? So we've got primary is basically sequence, and that's held together by peptide bonds. Secondary is going to be like alpha helices, beta sheets, that's held together with hydrogen bonds. And then tertiary is really bringing all of those side chains together. And so then we have all of the intermolecular forces that are sort of available for that. What's that? Primary is a peptide bond, and it's really the sequence. When we talk about primary structure, we're really just asking what's the sequence of the peptide. If you were to see a structure of a protein, like if we had, had a 3-dimensional structure, could you point to something that was an alpha helix, a beta strand? If you were to identify residues that would be found inside versus outside, right? And again, that's identifying and knowing, hey, W is something that's like a tryptophan, but, E is glutamic acid, and knowing where you'd likely find those, okay? Not too much in terms of the detail on hemoglobin function, only sort of as the application applied as we saw things in Biochem 2. So again, stuff from Biochem 1 is not like a deep dive. Even on Biochem 2, it's not really a deep dive. It's like the big picture stuff. So we talked about equilibrium shifting again, when we talked about oxygen binding, as it sort of related to thinking about how we manage blood pH and so forth, understanding the difference between these words, right? Oxidation is the change in the electronic state of something like iron, but oxygenation is the reversible binding of oxygen. So understanding kind of how those are different. I don't know what this is. Background items added. Somebody apparently is doing something on my computer. All right. We did revisit and see stuff with the Perutz model. So remember thinking about how we have those integrated fuel equations with kind of carbonic anhydrase chemistry, and then sort of the R state, T state of shifting. So the Perutz model was really understanding that T state does not bind oxygen, so it binds a proton. So T state of hemoglobin is protonated. And so being able to think about that in terms of how protonation state can affect oxygen delivery. So even though this is something we learned last year, I do want you to be able to still think about this because it's tightly integrated with metabolism. Carbohydrates, yeah, draw glucose. You are going to have to draw some version of glucose or be able to understand glucose derivatives, right? Like, oh, galactose is a C4 epimer, mannose is a C2 epimer. So if I can draw glucose as a Hayworth projection, then it's just easy to sort of switch what I need to switch to make it into galactose or glucose or mannose. Again, understanding the difference between a configurational isomer, we need to break bonds. Conformational isomers, I need to twist or rotate bonds, right? Understand how things are linked together. And then we can have a whole bunch of different nucleophiles because you could have an N-glycosidic bond or an O-glycosidic bond, depending on whether a nitrogen-containing functional group is your nucleophile or an oxygen-containing functional group is your nucleophile. Then with lipids, right? Be able to draw me triglyceride. If I gave you some information about a triglyceride to draw, you need to draw the glycerol backbone. You need to put on the fatty acids that comply with the direction that I've told you. Is it an omega-6 fatty acid, an omega-3, a saturated? You know, what's that information? And following those basic rules of cis double bonds. We've got no double bonds in conjugation, things like that. You'd be able to recognize the structure of cholesterol, understand that HMG, and I think I probably had this later, HMG-CoA reductase is that rate controlling enzyme. So any drugs that we've talked about, sort of understanding how they do their job. We talked a little bit about vitamin D, but not too much. That's just was part of this lipid chapter. This was a thread we saw a little bit, 2,4-dinitrophenol. So what is that molecule, right? That's that diet pill that discharged proton gradients. We really didn't see too much more about the potassium channel, so I wouldn't worry about that one, but we did talk about glucose transporters. So know the difference between GLUT2 and GLUT4, where we find them, how responsive they are, things like that. Again, we really didn't see much more with the sodium glucose transport system. But obviously understanding the different types of transporters. Is this facilitated diffusion? Is this passive mediated? And again, we saw a lot with the glucose transporters. So I would sort of have those glucose transporters in your head. You really don't need to do too much with catalysis piece because we really didn't do too much with that kind of moving on to biochem 2. I would know, like thinking about that big table, we know about 4 biological molecules, and we learned about enzymes that basically are related to all of them. So obviously RNase A is cleaving phosphodiester bonds, lysozyme is cleaving glycosidic bonds, serine proteases is cleaving peptide bonds. So be able to sort of have those connected pieces there. But really not too much in the details on catalysis because we really didn't go too much into that. You're not going to need to do the Mad Libs of like, diagramming an enzyme active site or things like that. Kinetics, again, you don't need to know deep dive on kinetics. I would understand the 2 important kinetics parameters, like just Km and Vmax. Vmax is how fast can you go? Km is how tightly do you bind your substrate? We talked about some examples of competitive versus uncompetitive inhibition. So being able to think about what those guys are. Nothing from a math standpoint. I think the Michaelis-Menten equation is one of those equations that we sort of have seen a little bit more. So I might know the Michaelis-Menten equation. And this is just sort of a theme that we've seen, how do we control enzyme activity? Right, know what covalent modification is. Okay, kinases and phosphatases. Know when those would be operational. Allosteric effectors change the shape from R state to T state. And we sort of talked a little bit about cytochromes again. We revisited them when we talked about heme structure, because cytochromes have hemes that are in them, but not really anything deep on kinetics. And then we really had an introduction to metabolism at the end of Biochem 1 that you're going to know sort of everything, because we elaborated on that in bio. That's all what Biochem 2 was. So don't need to, necessarily know this, but this is really sort of the high level that I would expect that you sort of know things. Think fundamentals. Go back and review your fundamentals quizzes. Know your HP axes. Think about a table. If there was a table for our important main HP, HPA, HPT, HPA, T, and HPS and HPG. Those were the gonadotropins, and then the HPA and HPT were the two main ones that we talked about. But then we also talked about the growth axis, which is the HPS axis, and then sort of the sex hormone axis, which is the HPG. Oh, I have them up here. HPT, HPA, HPG. Obviously, if there are some hormones, like you guys had to take a hormones quiz at the end of last year, the only ones that you really need to know are the ones that we've revisited. Obviously, insulin, glucagon. We've talked about cortisol. We've talked about adrenaline. We've had, a little bit of a discussion on melatonin, thyroid hormones. Those are sort of the ones that we've really spent time talking about. And that gets us through Biochem 1. So I'm going to pause there for a second. Anything you want me to sort of explain a little bit more? Lauren. So if you had to draw, like, what does H stand for? I'll ask you, what does H stand for? Phone a friend? Hypothalamus. What does P stand for? What's that? Pituitary. Sounded like you were saying pyruvate for a moment there. And then what is T? Because we've got HPT, HPA, HPG. Thyroid, adrenal, gonadal, okay? And be able to think about the hormones that are associated with that cascade, right? So for the HPA axis, it is adrenaline and also cortisol, okay? For the HPT axis, so yep, so we got T3 and T4, the downstream effectors. TSH is what's really secreted by the pituitary. And then HPG, we really didn't talk too much, but if you had to think about important, two downstream important hormones. Yes, estrogen and testosterone. We really didn't get too much into the LH and FSH sort of piece of it, which that's what's secreted from the pituitary gland, but sort of know sort of some of those high level things for that. Help, Lauren? Is that good? Other questions on Biochem 1 stuff? All right, so moving into 2. All right, so think fundamentals level, okay? Overview of the pathway. So you can see here quizzes. Again, be able to think about when, what would we be doing during alcoholic fermentation, homolactic fermentation? What are the main products? What are the main enzymes? Okay, so those are sort of the high level pieces. Obviously, we had a case study on metabolism of fructose. So that's kind of one of those other sugars that we need to think about sort of metabolism for. Know the main products of some of these pathways, okay? Not only NADPH, what else? I probably should have this here, but what else is important on the pentose phosphate pathway? R5P, right? And so when we had applications for things, we talked about G6PD deficiency, right? G6PD is the important rate controlling enzyme of the pentose phosphate pathway. So always be thinking, all right, if I pinch a pathway here, what happens, right? Stuff prior to it builds up, downstream of it is deficient. Glycogen metabolism, the sort of the big thing that we sort of had here was being able to think about what happens under high blood sugar or low blood sugar sort of scenarios. And that was kind of a theme that we carried through. So be able to think about the stuff that we've learned about and say, what would be happening with this metabolic pathway if I was fasting or feasting? Okay, and kind of be able to think about those sort of big level pieces. Obviously, the rate controlling enzymes of glycogen metabolism are glycogen phosphorylase and glycogen synthase. I should probably have synthase listed there. Signaling cascades, right? This is stuff that I do want you to sort of know. When we talked about our phosphorylation cascade, like be able to think about pKa, that's the R2C2 piece. Be able to think about phosphorylase kinase. That's the alpha, beta, gamma, delta piece. And then we talked about glycogen phosphorylase. That's the one that had serine 14 that was either buried or out. Okay, that is one of those that we saw several times. I do want you to know a little bit about the detail for how that cascade works. Okay. Think about the different shuttles we have, right? We talked about, many different shuttles. So we have the malate aspartate shuttle. We had, sort of the carnitine transporter. So just be able to think about the different ways that we know to get molecules sort of across. And be able to, as a way to quiz yourself, if there's something that needs to get between compartments, do I have a shuttle for it? Or do I need to, have a specialized kind of way to disguise it to get it across? And there's some things that we don't just transport without an important transporter, or don't transport at all, right? There's things like acetyl-CoA. Do we have acetyl-CoA transporter? No, like some of those really important branch point metabolites. Can I export G6P? Cannot export G6P, right? You have to do two things. You have to have G6Pase in order to be able to get it out. And you also have to have a glucose transporter that's there. So think about some of these molecules and whether or not they are exportable. Can I export and circulate ATP? ATP is used locally. Okay, I can't have like my liver make a ton of ATP and circulate that out. Now my liver can circulate out glucose so that other cells can go through their own glycolysis and sort of make their own ATP. So be able to sort of think about those high level questions, like, is this a molecule? And if we've talked about it a lot, do I know like the value of that in terms of what am I going to see? 5 millimolar is an important number. We see that for both glucose and for ATP. I don't have anything else I think I want to say with glycogen metabolism. And again, if you see something here and you're like, what do I need to know about that? Or I don't know this piece, feel free to step up and ask it. Again, citric acid cycle, pathway overview is really your fundamental space. Is that a question, Les? Yes.
There's 5 issues.
Yes. So, and I would say if you were to come over here and think about, I think we finally put all the pieces together in our glycogen metabolism learning unit, right? So, I probably have individual. So here's the pKa sort of like phosphorylation cascade. Then there's the phosphorylase kinase one, and then glycogen phosphorylase. So these 3 videos are going to be ones that walk you through sort of the structural details of those guys. So that is one that we went into some more detail. So does that help with that's what those are?
Yeah, would you ask that for like other one sides of like, it's just like it's a table. It has like a flow through.
Like on the homework?
Yeah.
So let me pull up the homework and we can take a look at that. And let me know if I flip by it and you see it, like, oh, like this one. So this one, yeah, obviously I'm not going to ask you the whole thing. It might be a part of it and it might be need state, fed state, things like that. But again, you can see these details are here because it sort of talks about the, what's being phosphorylated, things like that. So yes, but I'm not going to ask you that whole table. Yep. So keep in mind too, and I know I'm horribly probably bad at this, but I really don't want to make it something that your limiting factor is not having enough time to write something down. So I want to like see how well you can understand and integrate some information, but I don't want your hand to be hurting so bad at the end because you were just frantically writing because I asked so much. I don't know if that helps. So it's like it was before. Oh shoot. And you know what I'm just realizing here? I did not, as I'm talking in this video, you guys are seeing this, but I didn't share my screen. So I've been pointing to stuff. I don't think it necessarily, we're having a conversation and probably, I think it might just be Gabby and Colin. And are you studying with Gabby? No, maybe not. Okay. No, you don't have to. I didn't know. So, yeah.
She has questions she can ask.
Yeah, and she certainly ask me. What's that? Yeah, correct. So I'll share my screen right now. Let's go back here. Zoom. Oh, that is not a good look. I hope it hasn't been like stuck like that the whole time. Oh, that's really bad. Oh my gosh, somebody was gonna have to watch this because I'm not sharing the screen. And like them just stuck there and you gotta watch that while I'm talking. Oh my gosh. Oh, sorry. All right, so we were here. Maybe I'll come back. All right, so back to here. This is what we were looking at was sort of this homework thing. And prior to this, all we were doing, and hopefully you'd be able to sort of follow along, was going through this. So hopefully if you're, and I'll make sure to say something to Colin and Gabby directly to say, I forgot to share my screen, but hopefully you can get where I'm at by just following along on your own version of the study guide. All right, yeah, so I don't see my video face here anymore. All right, so we'll just keep it at that. Really very little with photosynthesis, so do not worry too much about that. Obviously, no Rubisco. I want you to know, like, that's a super important enzyme that fixes CO2, things that maybe integrate to other stuff. So as we had a compare and contrast with mitochondria, be able to sort of think about how it relates to things that we talked about with mitochondria. Yeah, the citric acid cycle, we really didn't even spend too much time on that because we did a good bit in the lab piece, but fundamentals, you had a citric acid cycle fundamentals quiz. That's going to be good enough for the citric acid cycle. ETOP, again, you had a fundamentals quiz on that, being able to think about like the importance of that proton gradient. And again, information on the mitochondrion as it relates to chloroplast. Yeah, I wouldn't even worry about plant adaptation pieces, you know, that was, you know, we're not even that high level here with photosynthesis. It's really understanding that photosynthesis is really a reversal of our key equation, right? So key fuel equation is reversed as we revisited it, and understand sort of chloroplast homology to mitochondria. Lipids, probably most current in your brain. The big piece there was, again, thinking about not specifically high versus low blood sugar as much as it was need state versus fed state, and sort of how we're going to manage what happens with either lipid breakdown or lipid synthesis. You know, we had obviously a case study on the ketogenic diet. So being able to sort of think about that sort of from a big picture standpoint. Had fundamentals quiz again on lipid metabolism. I think if you study your fundamentals quizzes, that's going to be a really good gauge for the level of detail I want you to know from these metabolic pathways. Okay. And then some of the big picture stuff, whether it was high or low blood sugar, need state, fed state for carbohydrate metabolism or lipid metabolism. Okay, those are sort of some big picture things. Protein metabolism, be able to draw an amino acid. Tell me about how it gets broken down. Again, you had that was your last fundamentals quiz was on protein metabolism. So look back at that. We had some applications for things here, PKU, MTHFR. We didn't talk about maple syrup urine disease, so you can X that one off, but we did talk about glyphosate. Just big picture, being able to think about sort of transport issues that we may or may not have. So think about those 4 biological molecules that we learned about in Biochem 1. It was proteins, carbohydrates, lipids, nucleic acids. And actually we learned about nucleic acids first in Biochem. But be able to think about, if I had a table that sort of asked you to relate and think about all of those, like what are they used for? How are they connected? What are the important atoms? Do we use it for fuel? What's sort of like the important biological molecule that we had for those? So that could be like a good way to just try to make a practice study thing for yourself, is could I put together a table that sort of thought about those pieces? And again, we had 1 day on nucleic acid metabolism. So really it's about uric acid. We spent a little bit of time sort of thinking about chemotherapy agents. So be able to think about, hey, DNA replication is really important for cell division, and cell division goes awry with cancer cells. Yeah, that's how we sort of tapped in to try to help with things there. Integrative metabolism, again, we really didn't have too much on this. You are going to look deeper into this, specifically on autophagy, with your take-home case study. And I wanted to make sure to say a couple of things here. Everybody has to submit their own, but you can work together with somebody. If you do work together with somebody, I want you to let me know who that person was when you turn yours in. Okay, just, Skylar and Kyle, you can be like, when you turn yours in, just kind of put who you worked with on it. Okay, but you can no more than 3. Okay, you can have 2 or 3 people that you can work with. In that case study take home, I think I said it was due Sunday, I think by like 5 p.m., let's say, or yes, because I don't really need to grade that. I just really need to look through it and see how extensive you sort of put time, effort, energy into it, and then assign sort of credit based on that. But yeah, I need time to grade the autophagy case study. So that one needs to be in a little bit early. Grades are due 9 a.m. Tuesday morning. So that's why I need a little bit more time. I need to have all day Monday to kind of be able to grade the other stuff. So let's see. There were some new molecules that we learned about with our integrative metabolism, and a few new cycles. So the big picture thing with this was really being able to think about how we have multiple organ systems talk to one another as they're doing their metabolism. So understand what the Corey and the Cahill cycle is. We had some new hormones like adiponectin, leptin, ghrelin. We really didn't talk too much about PYY, but I think it was on that slide. Some drugs that we talked about, metformin, that's again a drug we saw a few times. Know what that drug does, right? Who takes it, right? Who would take a statin? Who would take metformin? Like what would be going on, why they would take that? And again, your autophagy is going to be sort of the main focus of fasting and autophagy of your case study. We obviously talked about some important disease states. So diabetes, metabolic syndrome, and NAFLD were things that we sort of talked about. So not a lot of detail with that, but just understanding what these are and how they connect to things that we previously talked about.
Like there really isn't much on
I don't think there is. Yeah. Yep. And then again, none of this stuff, even though I listed here, you are not responsible for anything that we did in sort of our application piece. Again, anything that we talked about in the cancer metabolism case study, that was a revisit. Like I expect you to sort of understand the Warburg effect and why we'd expect there to be an increase in lactate production and a decrease in oxygen production. But beyond that, nothing on the lab testing. Other than some of the things we've talked about already, I do want you to know what normal blood sugar levels are for sort of a fasted state, right? 5 millimolar, which equates to 90 milligrams per deciliter. And we have talked about this a little bit, this concept of fat flux. So the deposit and withdrawal of triglycerides from your adipose tissue is tightly regulated by insulin. And that's why if you have insulin resistance, that sort of banking account, that was a little triggering for me to talk about deposits and withdrawals in a bank account right now. But that's why that is problematic, right? If you have insulin resistance, because once that's out of whack, your whole banking system is problematic. All right, so that's sort of running through this. I'm going to stop sharing right here. I'll see if I'm back. Stop and start video. See if I can. Oh, now I'm back. Who knows how long that like weirdo. This is like those weird places that you pause. Okay, so what I'm going to do, and I wanted to make sure I am not sharing anymore. I'm actually going to go, because that would be like really bad if, no, you guys probably wouldn't think it was, oh, running for school board. That's that. Now I have to unplug here. So I'm not sharing, but I was sharing my screen there. I am going to, I have to wait for my spinning wheel to stop. My computer's frozen. Come on. Now is not the time to kind of decide you're having problems. I literally wanted to take a second for myself, and I'm going to glance at the exam. And then I just wanted to make sure if there's anything else that I want to share with you.
So with the homework, like the question where it's shows like we start with glucose glycolysis and the Krebs cycle and all that.
Yep.
And you want us to fill in the arrows.
Yeah.
Like
I'm not giving you that figure that's on there and asking you to fill in the arrows, but
okay. I guess that's some reason I was thinking that would be that. I was just wondering what level of like detail would you be able to.
So for each arrow, like what is that metabolic process? Be able to think about like, okay, what enzyme is important for this? Is that arrow operating in a need state or a fed state? Like what's the starting and ending point for those? Like for glycolysis, I expect you to know that it starts at glucose, ends in pyruvate. PFK is the important rate controlling enzyme. Some other important pieces that we talked about that, like G6P, an important branch point metabolite that was there. Other than that, we really didn't talk too much about glycolysis in that regard. Okay, so hang on a second, and I'm just going to pull this up. All right, so you guys had asked about format. So it is 8 pages. So it's about your regular exams were 4 pages. You have 2 hours. So I don't think that's unreasonable. Okay. Front and back. So no. 8 pages total. So 4 pages front and back.
Oh, I was saying 16.
No.
I was just like, are we sure?
No. 8 total pages, which are 4 printed pages front and back. Okay, so there's a section here called structure affords function. Again, so being able to relate pH, protonation state, if there was a molecule that was there that you had to think about stuff. Okay, reaction mechanism. Know your functional groups and be able to think about biological molecules that have those functional groups. Okay. So like alcohol. No, well, like, so the functional group, there's 10 functional groups pretty much that we learned about that we saw. So if there was a ketone, show me like what, if you had to draw like ketone, R, carbonyl, R. Where did we see ketones? Tell me a biological molecule that we learned about that has a ketone functional group. I don't know. That was not see, I don't even know that because that's not a molecule. The question was asked, does uric acid? And I was like, I don't know. Pyruvate does, sure. Yeah. All of your carbohydrates have a carbonyl either as an aldehyde or a ketone, right? So be able to think about that. Your intermolecular forces. Okay, know what your intermolecular forces are, be able to rank them, and then give me an example of intermolecular forces.
Like we had like that little chart, and it was like in class last semester, but you had an example and then you had.
So if you had to tell me like Like you can either, you don't have to draw structures of molecules, but if you were to like, tell me, an intermolecular force. So if you had to tell me something where you see hydrogen bonding, okay, or let me actually see, it just says provide a biological example. So where do we see hydrogen bonding? You don't need to answer that now, but if you don't have it here, that's maybe something to think about, like, oh, where did I see hydrogen bonding?
Secondary.
Yeah, secondary structural elements for, or we saw it in holding hydrogen bonding in nucleic acids, right? We see that between base pairs. So be able to think about biological examples of where you see those guys. So I'm not going to like read to you what the whole test is here, but as I'm going question by question, I already told you this, right? There's a table here that highlights important features of 4 main biological biomolecules: carbohydrates, proteins, lipids, nucleic acids. Okay. So think about if you were to even try as a study guide to make a table for yourself, like what are things that we talked about that maybe we talked about for all of them, or maybe there's like a couple of cells that are blanked out because we didn't talk about that. Like nucleic acids aren't a food, so we don't have a calorie value for them. But what were some of the things that we talked about for those?
Is this part gonna look similar to the charts that you already given us on like exams and stuff?
Maybe, I don't know. I'm legitimately saying this. I don't know. We haven't seen this one because we haven't necessarily put all of this together. Okay. And then, so that gets us through first 2 pages. Then there's some questions about amino acids. Okay. And you need to give me single letter codes for them. I will give you one example just so you sort of have an idea, because the question says, what amino acid single letter code satisfies the following biochemical activity? Is important in forming disulfide bonds. But you got to give it to me as a single letter code. Yeah. Okay. So that's one example. Okay. There are 7 of them. Okay. 7 questions like that.
They're like polar non. Like. I do know, you're looking at.
I do know, but I'm not going to read you the whole test.
Like other fun facts about them.
Good study guide. Like try to think about.
Are we gonna?
I like that. That would be a cool thing. What's that? Yeah. So think about it too. I have to think about if I have your rapt attention for 120 minutes, what do I want you to spend your time doing? And it's not, you know, like I always would take issue with people like chemistry professors that would say you need to memorize and be able to tell me the entire periodic table, right? Like I had high school students who would tell me that that's what their teacher did. And I'm like, well, that's a lazy teacher because, oh, I shouldn't say this too much, but it's like, there's way better things you can do than have your students memorize the entire periodic table, right? Apply your knowledge. So, again, being able to look at a structure of a protein, and if I'm pointing to things, who is likely to live here? What kind of structure is this? Like, you know, what do you think about this? Drawing structures: carbohydrate, dinucleotide, tripeptide, and triglyceride, right? So be able to, given information that's provided, could you draw those guys? So it was carbohydrate. There's a few carbohydrates you have to know as Hayward's projections. Dinucleotides, right? And we never needed to draw the bases, but you need to show me where a base is attached, whether it's phosphorylated or not. Phosphodiester bond. Peptide, little peptide, that's a cute little word. And then triglyceride. Okay, so if you had information about a certain triglyceride. Then I've got a section here that's called physiological biochemistry, right? So some numbers, we already sort of talked about this. If you had to think about physiological parameters, right? What are some things that if we were to say, hey, what is physiological? Like, you know, we talked about temperature, pH, you know, physiological levels of important metabolites or things like that. Okay, so there's a short list of things on that. Physiological processes. So five questions on that. I think they're pretty high level, so I'm not going to give too much more about that. Then I have a section here called physiological markers. So again, if you were to, like, hey, this is a physiological marker for need state. Think your hormones or biological molecules or states or things like that that would put you into a certain physiological sort of situation. And then I told you about the HPX axis that was on your study guide. It's chart. That's chart. Yep, that's a chart. I have something here called experiment, and I have it in quotes, experimental biochemistry. So again, if you were to be looking at plots of things or things that are related to important things that you might see in a lab. Again, I told you the Michaelis-Menten equation, reaction coordinate.
Like one that we would have saw like last year.
Yeah, I mean, when we learned about, like, if you had to draw me a reaction coordinate, and I even tell you the pieces that you need to put into it, right? Like, here's all the pieces, and you could go back and look at previous homeworks or things like that, and you'd have this. It's like substrate product, transition state, what your delta G double dagger are for a catalyzed and a non-catalyzed reaction, and then talk about how enzymes work to catalyze reactions. We talked a little bit about SNPs. So even though we learned about them last semester, there was several of them that we sort of like talked about or revisited. So what is a SNP? And tell me a story about one. Okay. Math question. You're going to make a buffer. Tell me how to do some math, and then tell me how to do it. Yes. Yep. Obviously. Yep. I do not expect you to do math in your brain. We learned about a few key proteins or protein classes. So things like kinases, phosphatases, hemoglobin, myoglobin. Even though you don't need to diagram an enzyme active site, like, what are 3 things that when we do enzyme catalysis has to happen, right? How we regulate biochemistry. So again, that's some of those high level, what does it mean to be need state, fed state? What are some of the markers that we'd see for those? This is where, like, if there were drugs that we talked about and what they did, right? So we talked about statins and HMG-CoA reductase. We talked about metformin, and it's a first-line treatment against type 2 diabetes, things like that. Nope, that is like, there really hasn't been a free response one that's not a diagram, with the exception of like the SNP story, like define SNP and then tell me a story about one. Fill in the blank, like you've seen those like in your exams where it's like single word answers. Other than drawing a structure, mechanism, everything else has been a table or like short 1-word answers so far. Okay. Talking about transport, like if you had a table that had to think about the mechanisms of transport, facilitated diffusion, primary active, secondary active transport, and then we talked about uniports, symports, antiports, like be able to understand those, and then think about a place where we saw that. Okay. I have a section here called important molecules of biochemistry. These are single words, just kind of fill in the blank. I'll give you an example of one. These essential fatty acids are considered anti-inflammatory. These essential fatty acids are considered anti-inflammatory. Great. Yep. So that's an example of one, and there's about a dozen of those there. Okay, but they're not esoteric molecules. These are ones that we've talked about multiple times. Okay. And then I have one last question. Okay. And I'll tell you the, so this is the, it says complete the following table of the important things to remember about our biochemical pathways. Okay. So about all of the biochemical pathways that we've learned about, and some of these have like a spot that's grayed out because we really didn't talk about it. But think about your fundamentals things. How many steps was it? Where does it occur? What's the main reactant and the main product from the pathway? What's the main enzyme that we've sort of talked about with it if we did? So for example, in the urea cycle, we didn't talk about a main enzyme, right? But we did talk about some other aspects of it. And then if we talked about, is it considered a need state or a fed state pathway? Okay. So, and that is the last question. It's really asking us to sort of put together for our metabolic pathways, that information.
Is it gonna be like the other exams where there's like a list of, like how we had like the list of open-ended responses that we could, like how you had the, I guess the bonus on the last one. How you
Nope,
had-
there's no, no, I'm at the end here. It's done. Was that
I don't like
the open-ended ones?
I wanted to know if they would.
Nope. No, there's no, like, I literally read through the entirety of the test with you guys. Okay. Yeah, and I've got Gen Chem students that are waiting to come in and have their review. Yep,